Good morning. Thank you so much for being here. We're the Sharks research team. My name is Ellie Jester.
I'm Lila McCormick.
I'm Emily Ingebretson.
I'm Matt McGaffigan.
I'm Jesse Sweeter. I'm Sam Coles.
I'm Margaret Stoller.
So this semester, we've been assessing local shark assemblages in addition to coastal and international movements. So depending on species, sharks are apex predators who play a top-down role in their ecosystems to help monitor fish populations. Sharks are also keystone species, meaning that without them, there's potential for a trophic cascade. A trophic cascade is when the top predator, and in this example, the large shark, depletes, and then the next predator, the tuna in this example, would eat all of its food resource, the mackerel, and then both of those species would then deplete as well. This continues until the ecosystem is completely broken down. Unfortunately, sharks are very vulnerable animals because they mature slowly, reproduce late in their lifetimes, and only have a few pups at a time.
So historically, sharks have typically been perceived as dangerous and aggressive predators, leading the public to overlook their vulnerability. To give you an idea, about 6 people are killed each year from shark attacks, while over 100 million sharks are killed by humans. So humans pose a much bigger threat to sharks than sharks do to humans. Because of this misconception, until recently, few efforts have been dedicated to shark research and conservation, leading to a severe data deficiency across many shark species. This is a critical issue because without any information on these species, it's impossible to know whether or not we need to protect them, and if so, how we can best do that. So our research team is looking to fill some of those knowledge gaps.
Some of the problems that sharks face include exploitation as well as coastal development. And as you can see in the top picture, there are rows and rows of shark fins harvested from larger sharks. And so shark finning is a huge issue because of the high demand for shark fins, especially in Asian countries because of shark fin soup. Another issue that sharks face is bycatch, and as you can see in that bottom picture, there's a shark caught in a line. And so what bycatch is basically an accidental catch. And so that's a huge issue because it's hard to regulate. And another issue that sharks face is coastal development, which results in habitat destruction. So for example, shark nurseries are affected by that, and then that can impact the reproduction rates.
Sharks also suffer from data deficiency. Ever since 2011, the Bahamas has been a shark sanctuary. This means that there's no recreational or you can't catch sharks recreationally, and there's no exports or imports of shark products. As you can see, the shark sanctuary is a very large size. Unfortunately, there's not enough data to have sharks protected outside of the shark sanctuary. This is a past bull shark that we have tagged, and it shows that the shark migrates outside of the Bahamas where it's protected, past the coast of Cuba and towards Florida. During this time, the shark is not protected because it's out of the shark sanctuary, and it's very vulnerable to exploitation such as finning and bycatch.
So in this research, we're looking at the assemblages of shark species around the Cape of Eleuthera by viewing the population using long line. We are looking at the migratory patterns of bull sharks, Coccurunus lucus, by using acoustic and satellite telemetry. And we're looking at the migratory patterns of tiger shark, juvenile tiger sharks, Galiocerdo cuvier, by using satellite telemetry. We're using this data to fill any data deficiencies and to establish new protection areas.
This brings us to the methods of our study. So in our study, we use a long line, and our long line is 500 meters long and has between 40 to 45 gangins, and with a soak time of 90 minutes. Every 5 meters there's a gangin, and every 5 gangins there's a buoy. A gangin is attached to the long line with a rope and then a baited hook at the end, and the buoys are in place to keep the long line at the correct depth. This buoy. This is our sample zones. Here's the island school. And here's the Bahama Banks, also known as the Bank Zone, and here's the Exuma Sound, known as the Wall Zone. Each of these square boxes are our sample zones for our long line. Every shark that we catch goes through a general workup. So this includes first identifying the species of the shark and then measuring the shark. We take 3 measurements: the total length of the shark, the the precaudal length of the shark, and then the fork length of the shark. Shown in this picture, you can see Lila measuring the total length of the shark. We then sex the shark and take 3 samples. These samples include a white muscle tissue, a blood sample, and a DNA sample. These samples aren't currently used in our research, but are saved and used in research in the future dealing with reproduction and biology.
There are 4 tags that we use in our studies. The first 2 we put on every shark that we catch. There's a spaghetti tag, which is inserted just below the dorsal fin. And this has the contact information for CEI, so if the shark is recaptured by another group of researchers or even fishermen, they can contact us and we'll know where the shark is at that point in time. There's also a cattle tag, which is punched into the dorsal fin, and that has an ID number unique to the shark. The third tag is one that we use for any bull and tiger sharks that we catch. It's called the Pop-Up Satellite Archival Tag, or a PSAT tag, and it works by staying on the dorsal fin for a period of 6 months before popping off and transmitting all the data that's collected to a satellite, which then relays it to CEI. And this tag does a really good job of showing the large-scale migration patterns of sharks, where they could potentially be leaving the shark sanctuary and going into unprotected waters. The fourth tag you can't actually see in this picture because it's inserted under the shark's skin, but we use it for bull sharks. It's called an acoustic tag, and it works by sending a ping to receivers stationed around Cape Eleuthera whenever the shark swims by, thus giving us information on its coastal movement patterns. So the next couple of slides show some of the results that we've compiled from previous studies and our own. This graph shows the ratio of species and sex of sharks caught in summer 2016. So on the x-axis we have the species of sharks. There are black-nosed sharks, black-tipped sharks, reef sharks, nurse sharks, tiger sharks, and lemon sharks. And then on the y-axis we have the number of each species captured. And in total, 42 sharks were caught over the summer. And then this graph shows the same information, but for this fall. And we caught 23 sharks so far.
This results slide is showing 3 bull shark information of their depths using PSAT tags. And as you can see, 90% of the data has shown that bull sharks stay at 25 meters or less of depth. This results slide is showing local bull shark movements throughout Cape Eleuthera. Here's the island school and here's the marina. Within the marina, there are 2 acoustic receivers, making up 71% of the total tags, showing that bull sharks are most commonly found in the marina.
Currently we have 1 tiger shark. It is juvenile and it was caught on June 6, 2016. These are the measurements of the shark that we caught. We have not received data from the PSAT tag of its migratory patterns as yet, but the tag has a set time of 8 months.
So we have learned a lot about the local assemblages around Cape Eleuthera through our long line. So one of the main things that we noticed is that we have not caught as many black-nosed sharks as we have. So we caught a lot of black-nosed and reef sharks in the summer and not a lot in the fall. This could mean that there's species-specific migration patterns that we can look into in future studies. We also found that for nurse sharks, we caught many more males in the summer than we did in the fall, and we caught many more females in the fall than we did in the summer. This could mean that there's potential pupping migration patterns species-specific to nurse sharks.
Research tells us that sharks are more economically significant in a tourism sense opposed to a fishery sense. So that's why it's important to know where these sharks are located for the tourist attraction shark diving. And it's interesting to think about how sharks are not only ecologically important, but also economically important. And another thing to think about is that on our long lines, we have yet to catch a bull shark. So that could indicate that we are being selective with the size and species of sharks that we're catching. And so that's another thing to think about.
So far, for bull sharks, we have analyzed 2 different data sets. The first is time spent at different depths, collected from the PSAT tags of our 3 previously tagged bull sharks. We found that all 3 sharks spent over 90% of their time less than 25 meters below the surface, which is significant in a couple of different ways. First of all, it tells us where we as researchers should target these sharks in order to have the best success. In addition, this information should be communicated to commercial fisheries as a depth range to avoid in order to hopefully minimize bycatch rates. And third, it gives us more information on bull shark habitat and behavior, specifically temperature ranges, species interactions, and more specifically, what species these bull sharks might be preying on, all leading to a better understanding of how these apex predators are affecting their environment. The second set of data that we collected was from the acoustic receivers placed locally around Cape Eleuthera. We found that over 70% of the pings occurred within the marina, likely due to the fact that fishermen regularly clean their catch there.
As previously stated by Jesse, we are still waiting for the data retrieval of the one juvenile tiger shark that we have had tagged. We also hope that throughout this semester and throughout the rest of this research project, we'll continue to tag more juvenile tiger sharks to learn more about their migration patterns. We also hope to do similar studies with other species to compare them to tiger sharks and to bull sharks, which we're doing now. And we also hope to continue with our longline survey so we can compare population trends to other species. In conclusion, we hope to bring more awareness to this amazing species and this amazing animal so that they will be protected in the future and not feared in the waters. Lastly, we'd like to thank the Island School, CEI, the interns, gap year students, our parents, and lastly, our research advisors, Heather and Maggie. Unfortunately, Heather could not be with us today, but has played a huge role in our success. This is our works cited page. Are there any questions? Yes?
What was the most unusual or surprising finding out of everything that you gathered?
So the question was, what was our most surprising finding? And for me personally, when we were out in the field one day, it actually wasn't a shark, but we caught a big fish head. We're not sure exactly what animal it was, but the fish head had been bitten off the body, and so we're thinking that's obviously a shark that came there. Does anyone else want to answer that? No. Any other questions? Yes.
So, based off of when you got the tags on the sharks and tracked them afterwards, were you able to form any hypothesis on the effect of the long line fishing on the sharks and whether it was all damaging to them, both directly after and long term?
So the question was, do we know if there are any effects on the sharks that they have from longlining? And we do assess that to the best of our abilities. After we remove the hook from the shark's mouth, we rate the condition of the shark on a scale of 1 to 5, kind of telling us how the shark swims away, and that might be a clue to how they are dealing with the stress of being on the long line. And from what we have seen, there hasn't been significant effects to the sharks. Yes?
The question was, do we actually have to go out and retrieve the PSAT tags? And we don't have to. The data gets relayed to a satellite, then transmitted to a computer. So we have that there, but due to bad weather, sometimes not all of the information gets to the computer. So the best method of retrieval is actually going out and finding it, but that can be challenging sometimes. So, yeah.
So your findings, the acoustic things that you created, findings show that bull sharks are going to the marina. Are there any efforts underway to change the behavior of fishermen about putting their fish there in marinas? Obviously, as human-induced changes to the patterns of bull sharks that would normally be there, are there any efforts underway to change that?
So the question was, is there any efforts going or underway to maybe protect the marina from bull sharks? So you're not actually supposed to swim within the marina, so there isn't a threat to humans. So fishermen, they clean their catch and they put the catch in the marina and it causes the bull sharks to come. That doesn't necessarily mean that any humans are in danger.
Yeah, actually, not as much about humans, but more about changing the behavior of the shark. That's not sort of a natural environment.
No, it's not a natural thing for the shark to do, but it is possible for, we don't really look at the food source of the shark, like as it's getting food, but we're going to the marina and we know that the sharks are in the marina, so we can collect samples from them. There are no behavior changes underway though.
Will any of the sharks that you caught, sharks that had already previously been tagged?
So the question was, any sharks that we have caught, have they been previously tagged? So yes, this semester we actually caught one shark that CEI had caught in the future, or in the past, and and we knew that because of the spaghetti tag, and it showed us the ID number of the shark, and we went and looked it up, and we found that it was caught in years past.